We want to welcome you to STEMaway, and we want to welcome you to the field of bioinformatics. It's an extremely interesting one. I'm assuming the people on this call are new to bioinformatics. This will be maybe your first steps in this field. You may have heard about the impact of bioinformatics. What you'll get to see over here is exactly how things are done. And it is challenging, but it's very thrilling because bioinformatics, you get to work with data. There's a lot of computer science involved. There is big data analysis. And at the same time, you're also working with biology. The impact of all of the analysis that you're doing is directly in cutting edge breakthrough moments in medicine. In STEMaway, we have several pathways. Bioinformatics is one of them. For me, my background is in tech. I'm the founder of STEMaway. The remaining pathways that we started with are all things that I had some experience with. Even though my background is in hardware, I have a lot of breadth, so all of these pathways I was familiar with, except bioinformatics. In summer, I spent every second that I could get out learning along with the rest of the students, and I really enjoyed the process. I also got to know the student leads, and so it is my extreme pleasure now to welcome Anya. She was a project lead in the July session, a participant in June, and now she is going to be the mentor for the Pathway Hub. And you are in really good hands to take your first steps in bioinformatics. So over to you, Anya.
Hi, as Marina said, my name is Anya. I participated in both summer sessions in the bioinformatics pathway. I have a background in data science, actually, but I've worked in an evolutionary genomics research lab applying data science in a bioinformatics domain. We're going to go through this presentation, and then I'm going to show you an R demo, and then we'll talk about the logistics of the project and the Pathway Hub. This is just a little bit about me. As I said before, I have a degree in data science and worked as a research assistant. I've worked mainly with repeat genetics, so like repetitive DNA and satellite sequences and whatnot. For the bioinformatics Pathway Hub level 1, this is our project definition. We just want to introduce students to bioinformatics and the main pipeline used in large-scale data analysis, as Debelina said. We'll be starting with introducing you to the basics, which is getting you a good foundation of R and R programming, and then we'll move into the actual pipeline, starting with an introduction to the data visualization and quality control, and then moving on to statistical analysis and functional analysis, where the biology really comes in contact with the coding. And lastly, I don't know if we'll be doing this for the Pathway Hub, but for the summer sessions, we worked on a capstone project, which was essentially applying the pipeline that we learned in the previous 6 modules to a new data set and sort of like looking back on where we started and where we are now. So a quick introduction to bioinformatics. It's the intersection of statistics, computer science, and biology. And so there's a lot of different facets of bioinformatics, and it's a very large field. As I said, I worked in repeat genomics. You can also work in normal genomics and transcriptomics, and there's a lot of applications of bioinformatics in general. For the pathway, we'll be focusing on the computer science and the biology, and using packages and functions to do the statistics. A typical bioinformatics research pipeline starts with data collection and data wrangling, and then we'll be moving on to exploratory data analysis, including visualization, quality control, normalization, and then you move on to statistical analysis with differential gene expression, and then finally functional analysis, where you figure out the functions of the statistically significant genes that you found in the previous step. You'll notice that this pipeline matches the module outline that we have planned. Back to the data collection, there's a lot of different ways that you can collect genetic data, and the data that we'll be using was collected through microarray sequencing. And the way microarray sequencing works is that you have a 2D array, a chip, and each column is a different sample, so it has DNA from a different tissue sample, and each row has a different complementary DNA sequence which we call a probe, and it's related to a specific gene sequence that you're looking for. And so you mix the sample with the probe, and the DNA hybridizes, and with different fluorescent markers indicates a different expression level, and then the chip is scanned to turn those expression level, or like the fluorescence intensity, into a specific number. And this is represented very similarly in a data frame, with each column being a different sample and each row being a different probe ID, and the numbers representing those expression levels. We'll be, for the bioinformatics pathway, we'll be using a lot of different packages. And so it's important to understand how to install packages. Typically, you would use the install.packages with the package name for a normal R package like ggplot2. However, we'll be using Bioconductor, which is kind of like a package hub and a package manager, to install other packages such as Enhanced Volcano and AFI and AFI PLM. And so it's just important to note that they have a different install method, which is bioc manager :install and then the package name. One thing you may see come up when installing packages is to use this if statement. And this basically says, if the package is not already installed, then install it. So when you write an R script, you typically run everything in the R script, and when you do that, you don't want to have to install the package every time you run the script. So that's just why you see the if statement. Another important thing to understand when it comes to packages is reading documentation. This documentation, which often looks like this, it has a description, an example of the usage, a list of arguments, and then some notes and maybe some examples on how to use a function. So you can find this documentation in RStudio by using a question mark and the function name, or you can go to the R documentation website, and it has all the same information as well. When looking at documentation, what I tend to look at, and what usually provides the most info, is the arguments. The arguments tell you essentially what the function can do and what each parameter can be. For example, for g_on_point, which is used in ggplot to make a scatter plot, you can give it arguments for the data, the mapping or aesthetics you want it to use. And those are the two main ones. Yeah. If you ever have any trouble reading documentation or want to learn how to read it, just reach out and I'd be happy to help you try and understand it. Another thing to note is the bioconductor documentation, which is slightly different from normal R documents. So if you go to the bioconductor page for Enhanced Volcano, which is another plotting package, it helps you plot enhanced volcano plots for differential gene expression. So the bioconductor page has a description, has instructions on how to install the package, and here you'll see the if statement that I mentioned earlier. But what's most important on this page is the documentation heading, which includes a reference manual typically, which is the typical R documentation. And it oftentimes also includes other links to other helpful resources. For instance, this HTML page is a walkthrough made by the creators of Enhanced Volcano, showing you how to use it in different use cases. Another package that we can look at is AFI PLM, which is used in quality control. So if you go to the AFI PLM bioconductor page, it has a bunch of PDFs, and each of these PDFs is for a different use case of the AFI PLM package. For our purposes, we'll be looking at the model-based QC assessment. And you'll get more information about this in the coming modules. But specifically, we'll be looking at RLE and NUS plots. And so there's a ton of information in this PDF about how to get those in R and how to interpret them. What I'm trying to say is that the bioconductor documentation is very helpful in not just understanding how to use the functions, but also how to understand how to interpret the output of the package. Lastly, you can always look on forums. You can ask on the Semway forums, or you can look at Stock Exchange, and BioStars is a bioinformatics specific forum. It's important to note when you're asking questions to include the code and the data you're using, and also try and include like the specific issue or the error message that you are getting. Okay, are there any questions about that? Okay, I will move on to a quick R demo, sort of walking you through the first few steps of the pipeline. So this is my R script, and these are the libraries that I've loaded, or the packages that I've loaded into my environment. For our purposes, we'll be using, you've heard in the R training, you've heard about like read.csv and read.table to read in data. However, the data that we'll be using is in a different format that isn't as easily readable, and it needs to be translated to be a data frame. So we'll be using the function read_affy for that. And then for quality control, one of the many quality control packages that you'll be exploring, but the simplest one is simple_affy, which uses a function called QC on the affy data, on the affy batch data that comes from read_affy. And then you plot that output and you get this plot. And this is basically showing you which samples are outliers or which samples have a different quality to them. If you want to know more about like this specific plot, you can always just type into Google the function name and the package. And typically you'll find something like this PDF, which has, if you scroll down, has a like point-by-point diagram of how to interpret this plot. And one of the reasons it's important to like understand how to look up what output means and stuff is that in this pathway, we'll be using a lot of different packages with a lot of different output. And so it'll be very hard to make an exhaustive list of how to interpret everything. Next, the next step would be normalization. Here we're using RMA. There are lots of different normalization methods, including GCRMA and Mass5. RMA runs the quickest on my computer, so that's why I'm showing you this one. Normalization is important for making the data comparable. If you'll see in this box plot, this is the normal data where each box plot represents a different sample. So you can see that all the samples are quite different, but if you normalize the data, they become comparable. Lastly, I'll show you two of the visualizations for the data, the PCA. This is a PCA plot. And PCA is a method that reduces the dimensions of data so that you can actually visualize it nicely. If you've ever done linear algebra, PCA uses eigenvalues and eigenvectors and bases, and a lot of different ideas from linear algebra to justify the lower dimensions. So typically in PCA analysis, the first principal component, PCA stands for principal component analysis. So the first principal component and the second principal component, typically, if the data is high quality, will represent 90% of the variation in the data. And so essentially what this plot is saying is that all the cancer data based on the first principal component and the second principal component cluster well, and all the cancer data is like similar and all the normal data is similar. Another visualization that we'll be using is the heat map. So this is a heat map of negative correlation. So boxes with a yellow or blue color show a lower negative correlation, meaning a higher similarity. And boxes with red show a lot of dissimilarity. And this heat map also uses hierarchical clustering, which is essentially just like a family tree. Samples that have a closer relationship at the bottom are closer related. And so this is just showing that all the cancer samples, which are blue annotated, are more similar to each other than they are to any normal cancer sample. Okay, that was a lot of information all at once. So if you have any questions, please feel free to ask.
That was a great summary. And as you said, it's a whole lot of information, but the idea is to give the audience some glimpse into what you'll be doing. Each one of these modules will take you step by step through the demo that Anya just did. Maybe today you don't have a question, but when you are going through the modules, let's talk a little bit about how the program is set up. You could kind of think of it as an extended training for the next virtual internship session. Although for people who attend each and every module, we do have a certificate of completion for the Pathway Hubs as well. In the internships, everything is done in teams. In the Pathway Hubs, it's more of a flat structure. So everyone in bioinformatics in level 0 and 1 will be working directly under Anya. If you have been on the forums, you have seen that she has already posted the first module. That's where all the information is about what is the first step that you need to do. Goal of this webinar was to give you a glimpse into what are the next steps and what is the overall project that you'll be working on. STEMaway is a forum-based platform, and the category that is of interest to you, the channel that you should be looking at is under Pathway Hubs is bioinformatics. That's where all the information is. And there's also another channel called STEMcast, which also has a sub-channel of bioinformatics, where all the resources are. So there are a lot of resources on the web, and what is unique about STEMaway is whatever resources is there in the STEMcast is directly resource that we're using for the project that Anya just went through. Very briefly, that is the overall structure. In terms of logistics, for every module and people who are following along, feel free to ask any question that you want to ask on the forums. We are monitoring the forums. Anya is there to help you. Other people might also jump in. We have many students from the summer session who have gained a lot of knowledge. And then at the end of the 2-week, we are planning for a 2-week session for every module. If people need more time, we can extend it. You have to submit a self-assessment which shows what you have learned and kind of captures the hurdles you face, the open questions that you still have. And then we'll have a follow-up meeting after every module where you get directly to ask your questions. And it's Anya is there to give feedback. We may invite some other people in, also other experts in as well at certain times where when students are ready to give their presentations, there will be an additional audience as well. What we found in the virtual internships is that feedback that you get from your mentor is very, very useful. So these meetings that we are planning at the end of each module is where you will really hammer in your learning. And it's feedback from Anya, and it's also, you can think of it as a brainstorming session where you ask your questions. Maybe you come up with some questions that we haven't thought about before, maybe some angle you see which we didn't cover in the modules. So it is a Q&A, but it's also kind of like a brainstorming where you're continuously working on the project. Anya, do you want to add anything to that?
No, I think you said it perfectly. I'm always available to answer any questions that you have.
All right, let's wind up today, but again, any question, come to the forums. If there's a question about logistics, reply to the topic which says the instructions for the bioinformatics pathway. Make sure that you've joined the bioinformatics group, because then you will have the required permission to reply to those topics. If there's a question about the module itself, about the technical content itself, then look for the post that Anya has done on the level 1 module 1, and that's a place to be. So welcome once again, and hope to see you on the forums. Thank you so much, Anya.
Thank you, Develina.